Okay, I will start all over again. Today I'm going to try to present Today I'm going to present a research work from um, Fransky, Bell, and Gansky, and Bexnovic's labs innovate articles about Cas9 VPR system mediated transcriptional activation. So generally, in this study, they try to express conopsin into rod photoreceptor. So after this treatment, the rod photoreceptor can express the conopsin and gain the photoreceptor functions. That is, it can be activated or functional in a phototopic or daylight conditions. This transactivation approach can activation a very large gene sequence expression in a cell, and it can be approached to cure some disease that was an autosomal dominant inheritance. And this system can also treat some very complex retinal disease, including multiple genes. And this treatment can be more popular than other editing approach because it's a mutation independent. It just directly activation another gene in a patient's cell. So this can allow for treatment for a large number of patients. So the original CRISPR-Cas system is a gene editing. That means this system coming from eukaryotic um, um, biology stuff. It's including several proteins form a cascade complex and they binding an RNA. And then this RNA can recognize and that's the same sequence in the genome of the target cell, then binding the whole cascade complex into that site, and then do the editing that's with the template of this RNA. So in this way, it can do the gene editing. But in this study, they're not doing the gene editing. They use this Cas9 system because it can bind in RNA, then only for recognition of the target genes, upstream promoters or site. Then it forms a chimera to bind in with an activator. And this activator in this system is VPR, is VP66, P65, and RTA complex. And this fusion protein fuses again onto the Cas9 system. Then they can bind into the gene's upstream sequence, then attract an RNA polymerase to bind into the promoter of the target gene, then start the transcription of the gene. And this attraction may be due to like a protein-protein interaction. So for most of the gene therapy, it's using the AAV system, but the capacity of the AV system is very limited. It's a high limit. It's only like a 4.5 KB. This is the whole package, but this Cas9 system is quite big. So you have to like split the sequence into two parts. So it means that it requires a dual AV vector that coming into the same cell. Then within the cell, one vector express an N part of this Cas9 system, and another AV construct express the C part of the Cas9 system. Then these two parts combining together, then they form the intact functional machine of this Cas9 system. And this Cas9, this binding is due to because in middle linked with a protein is called intent, and this intent will be cut off, and then this is the whole intact device binding to RNA, then recognize the genome sequence, then start the activation of gene transcription. In this study, their vector design is like this. One of them is an U6 promoter to drive the single guard RNA and 3 sequence, and use human rhodopsin promoter to drive the Cas9 and sequence. And another vector is using human rhodopsin promoter to drive the C part of Cas9 system and the VPR, the protein complex. So these two sequences come into the same cell. Have to be coming into the same cell, then start like form an intact a whole Cas9 VPR system. But from all of this study, they're only using human rhodopsin promoter. But in all of the study, they're only either like cell lines or mice. So actually, I'm not quite sure that why they use human rhodopsin promoter. So maybe it's better to use mouse rhodopsin promoter. We get better results. But if they can get it, so maybe that this human rhodopsin promoter is also works. So the target gene, they're trying to activate expression, they're choosing the first one and the last one in a light signal cascade. The first one is the opsin, and the last one is the CNG channel. So for the light transducing process is that the opsin can activate the G protein transducing binding to BDE6. Then BDE6 will start to hydrolyze the cyclin GMP, then cause the cyclin GMP concentration decrease. And this decrease will close this cyclin GMP gated cation channel, CNG channel, and close then the photoreceptor hyperpolarize and the transmitter release is reduced. So in this study, they try to put the con opsin into the rod photoreceptor and the con cation channel, CNG channel, into rod photoreceptor. The two different composition of the CNG channel in rod photoreceptor and comparing to the cone photoreceptor. Rod photoreceptor is A1B1 and the cone photoreceptor is A3B3. So in this study, they express cone CNG A3 into rod photoreceptor, but in study, the sequence they're using is the CNGA1. Actually, I'm a little confusing. Maybe in a rod photoreceptor, this CNGA1 is not from cone. Rod photoreceptor, express or upregulate its own CNG channel to see if it's functional. Well, another pretty cool technique they're using in this system is the PB transport system. In this way, they can establish a very stable Cas9-VPR cell line system. And so to generally say, it's like transfect the plasmid, and also with the transference, it's like an enzyme that can cut the target sequence from the vector, then insert into the genome of the target cell. Then in this way, it can insert the whole. Then in this way, they can generate a stable cell line. So in this study, the cell line using is 661W photoreceptor cell line that coming from the mouse photoreceptor, and another one is MEF, mouse embryonic fibroblast cell line. Another one is the HEK cells. The animal they're using is a 10 C57 mice and a 10 rhodopsin heterozygote mice with the background, with a genetic background, same as the C57 wild type mice. And so it's a 10 to 10, and these animals are age matched. I checked a review talking about the difference between the rhodopsin heterozygote against rhodopsin homozygote. And so comparing to rhodopsin homozygote, rhodopsin like a totally knockout, homozygote, and the photoreceptor auto segment is majorly is still preserved, although it's shorter than the wild type control. And the cell and the autonuclear, the photoreceptor cell density, not density, the total amount of these cells is reduced, and the thickness of the ONL layer and is thinner than the wild type control, but it still have a preserved an intact outer segment, the shape, but the homozygote is totally just gone. But and also we can compare the gene expression level between the homozygote and the heterozygote, and we can see and this data is normalized to the wild type. So so the number one means how to say so 两只 the number one means that the amount of gene expression is the same as the wild type. And so we can see here the gray bar, the heterozygote one, actually its protein expression level is not dramatically like reduced comparing to the wild type control. The only like dramatic growth one is a transducing alpha subunit, and another one is a arrestin, but it looks like it's pretty good to me. But the other for the homozygote, the white bar it seems it's really like dramatically like affected by this mutation. So how much does this heterozygote is functional will be worse than the wild type? And if there's no big difference, how do we know that this study can dramatically improve and give a solid prove that this treatment works? They also use other method like RT-PCR and subretinal injection, and it's a single subretinal injection. Inject 1 microliter into the eye and subretinal on the, postnatal 15 days old. And they also use patch clamp and OCT and ERG. So for the ERG data, they both use an scotopic ERG that is dark adapted and light adapted ERG. And the way A wave of the ERG that represent the response of the loud and comfort receptor, and the B wave is dramatically respond to Ohm type bipolar cells response against the light. Okay, now let's move on to see the results of this study. So firstly, let's try to prove that their system works in the photoreceptor cell line, and for separately they express the CNGA1 sig they try to activate the expression of the CNGA1 channel, and this is the site shows that where they choose the binding sequence of the single guide RNA. And another gene I try to express that separately. That at the a cell line experiment stage, they didn't try to like put all these two together, so it's a separate. So the sequence recognize the upstream of the upstream. It's showing here, three sequence. So in the photoreceptor line and with this Cas9-VPR system, there's this channel expression level increased, but and there's a no background expression because the control using the like this sequence cannot activate the expression of the CNGA1 channel. And in another experiment, they show that the expression level of the upstream medium wavelengths is also can be increased. But we need to take note that here is a relative expression level. So actually, let me see where. Let me see. Oh, okay. So here. So in this supplementary data, and we can see that there's a background expression for the Lexi sequence. So it means that this photoreceptor cell line have a background expression level of the opsin of the opsin. So this cell line looks like a mixed. It's not like the specific rod photoreceptor cell line, but it's like a kind of a mix with some like Bohm photoreceptor functional protein. That's the mid wavelength opsin. So here there's a background expression level. So, and that's the reason why in the next step, let's switch to the mouse fibroblast cell line, the MEF cell line. Let's go back. So, in addition to totally with or without expression level, they also tried with an inducible system. And this inducible system is using TAT receptor and the DOX inducible system. So generally say that the expression level of the dCAS9-VPR system is be controlled by the TET receptor system. So after adding the DOX, then TET receptor will bind into the promoter of the dCAS9 system, then start to produce the Cas9 VPR expression. And this expression is dose dependent. Kind of like a linear response against the DOX concentration. However, the functional in expression activation of the CNGA1 and have a optimal dose that is a 5 nanogram per ml, and without without the DOX, there's still some this CNG channel expression. So that means that this dCAS9-VPR system is not tightly controlled by the inducible system TET system, but if there's a no this ligand, there's still some unactivated dCAS9-VPR system to produce CNG GA1 protein. So this is described here. Then because there's a background expression level of the opsin in the photoreceptor cell line 661W cell line, so let's switch to MEF cell line to try to induce the opsin expression. It's pretty good because if there's a no DOX and there's a no expression of opsin, so this is tightly controlled. And the same as the CNGA1 expression, the 5 nanogram per ml concentration of DOX can produce the best results to produce a targeted gene. Although the Cas9-VPR system itself is expression level is linear corresponding to the DOX concentration. So in here we can see that maybe too much like a Cas9-VPR system can cause some like toxicity or something that can induce like the reduce of the expression of the target gene. So here's an image show that to demonstrate that in the HEK cells, and this system is also work for the control sequence from the Lexi. Let's know this CNG1 expression, but for the positive one that they using the single guard CNG1 sequence, and there's a positive transactivation of this CNGA1 channel in the HEK cell. And the HEK cell endogenously do not express this CNGA1 channel. So here is a patch clamp recording data from the photoreceptor cell line, and they're using a voltage clamp then to activate the cells while changing the holding potential or clamping potential. So at the rest state, the holding at 0 microvolt. Then first they do hyperpolarize. Then we can see it can cause a CNG channel, not cation, means a cation influx into cell, then holding to a positive, then cation flows out of the cell. And if using the magnesium, and this system will be blocked. And also without adding this cyclin GMP, because cyclin GMP is to ungated the channel and is also calcium dependent. So here we can see that the current with this Lexi sequence, it means that there's no this channel expressed and the current is 0. And this current can be totally blocked. So it's a clean cyclin GMP gated current. And with this transcript activation system, and there's a current expression. So there's a current shows, and IV curve is show like this and shows a cyclin GMP induced current. So the next step, putting the system into in vivo. So first step, protein on the Y-type mice. In the Y-type mice, there are several photosensor like a cone opsin. Of course, there's a short wave and a blue opsin, and rhodopsin on the rod photoreceptor, and melanopsin on the ganglion on the IPRGC ganglion cells, and activation a system that try to express the cone opsin. I marked the red here. That's this like a lot photoreceptor have both cone opsin and the rhodopsin. And here, because they have to use the AAV uh, um, transfection system and using human rhodopsin promoter. So this dual AAV vector design is like this, that one construct have the single guide RNA sequence, and part another is C part and the VPR fusion protein. And 4 weeks after injection, and we can see that there is an increase in the of the opsin middle waves in the treated retina. So here the sequence using this RNA sequence, they're using M-opsin or S-opsin. So this is the first from this data from first animal, and this data from the second animal. The endogenous expression of M-opsin we can see here is like this, but after treated with the dCAS9-VPR system, because there's a lot of there's a cone opsin expression lot photoreceptor, and the rod photoreceptors auto segment is a really long, and the cell number is much higher than cone photoreceptor. So you see that there's a totally super high expression of opsin in the outer nuclei in the outer segment there. And in the figure E and F, and they using another sequence that is the short wavelength opsin as opsin expression. We can see like this, and then it also can be increased by this dCAS9-VPR system. So it demonstrates that in Y-type mice this system works, and there's the cone proteins that expressed on the rod photoreceptor of the segment. And next, let's move on to see the functional data of this system. So here it shows both the dark adapted the scotopic response and the phototopic response. So okay, figure A is about the anatomy condition of the retina. The outer nuclear layer, that's the photoreceptor soma located, is the thickness is increased after the treatment compared to the saline control. Although you cannot go back to like the same as the Y-type. Oh, here because it's functional data, so it's they're using in the rhodopsin heterozygote mice. So it's red data and shows that it's not like matched to the Y-type mice, Y-type retina. And firstly, let's take a look at the phototopic A wave. And so generally the result are quite close, and with the light intensity increase, there is a wave's current. It's getting larger, and I need to point out that here at the very bright light condition, the response of the photoreceptor is even stronger than the Y-type control or saline control. The Y-type positive control and the saline the negative control. And And the B wave, that's the on bipolar cell response are also like improved. And there's no like a super like a super like surpass the Y type response, but it's still improved in a scotopic response, better than the saline negative control. So this response of the treated is like just between the response from the Y type control and the saline untreated negative control. But we can see here that the amplitude, this like difference is really tiny. But when we take a look at the scale of this, then we can see that actually this like the 5 microvolt change was really small. So although in this figure that shows that like the zoom in, I can see the difference, but this difference is not very big. So when we take a look at this data, then we zoom in at this part, it's a really tiny difference at here. In the supplementary data, it also shows all the 10 mice response, like one eye is treated, another one is the negative control. And when we take a look at this response, actually, still, there's no dramatically difference. So, well, we cannot say that this system does not work, but maybe this rhodopsin heterozygote is still preserved a pretty good functional. So the improvement is not so like obvious. So the next step, they want to see that if there's some kinds of like toxicity, immune response, or gliosis happens in this treatment. So they use like PRPH2 to stand the rod and cone outer segment. And of course, other like PNA is stand photoreceptor to see that the whole like the thickness is preserved, and there's a lot of cone opsin in expression level increase, and there's a little increase in other segment, but although here we cannot like judge that is increasing very dramatically with our naked eye. And the next step, let's use an IBA1 to scan the positive microglia or mononuclear cells to check if there's this if this like immune response cells penetrate and into the photoreceptor layer, because this response, if it's too high, it will cause the damage. That's a gliosis, and that's a pathological conditions. For example, let's take a look at the RD1 control, and this microglia and mononuclear cells, the blue one, is there's a lot of them and that penetrate into the photoreceptor there at P14. And there's a very activate, like Mueller cell, glial cells that's in the retina in the of the white RD1 mice. But after like 1 years old mice, for these treated mice, the retinas looks pretty normal like this. So they also like check the apoptosis, like how to say 程度 apoptosis like a condition and the control I choose the P14 RD1, and there's a lot of this is used in children studying, so there's a dramatically like apoptosis that express that show up in the RD1 retina. So that means there's a very active like photoreceptors dying in the P14 of the RD1 mice. But for comparing to the treated or untreated, let me see. This is a treated and this is the untreated um, retina, and the tuner positive cells is less than untreated one. So here they claim that it's like um this treatment can help the retina be more healthy to help the photoreceptor survive more. But um, I still have a little critique here because this is 1 years old animal. I think that is already quite long, but for the let's see, it's like for RD1, that's a very young animal. That's just 14 days old. If like we see, because they do injection at the P14, so everything happening. I think like maybe like 2 weeks after the injection, or like 1 month after injection to examine the ptosis condition. 1 years old is really too long because 1 years old those cells dying already die. It cannot be stand by the tuner. The cells just the everything is being removed by like other like um microglia cell, those that immune response cells. So here image is a higher magnification image to show the outer segment that is indeed this on opsin and opsin is present in the rod photoreceptor because it presents a very long outer segment. So the conclusion of this study is that um, this dCAS9-VPR system works, and the CNGA1 channel and RPN proteins can be transactivation to transient expression in photoreceptor cell line and fibroblast MEF cell lines. And TETR and DOX induced system can also express the cause inducible expression in a cell line system. And in vivo, this split Cas9-VPR system with the link of the protein intent can still functional to activate the gene expression of the CNGA and OPN1. And it works in wild type mice. And this treatment can delay retinal generation and improve retinal function. They also present data that will reduce apoptosis and without induced gliosis or invasive of immune response cells. So gliosis and immune responses is quite persuasive, but reduce apoptosis, maybe, but um, the data point, the time point they're choosing 1 year old make me still feel. So and the last they also provide experiment data about that the saline solution treatment is safe. It's quite safe. It's not big difference comparing to completely without injection those untreated rhodopsin zygote retinas. So for here, for both gene considered from lower than. And in the discussion part, the authors talking about that this split system, well, it's like very like realistic restriction because that it just cannot package this too big sequence of Cas9 system into one AV. So they have to use the split, and it required like two virus enter into the same cell, so that transcription efficiency is lower, and this is discussed in this article. So the controller using white type mice, I still have a very picky question. Why they using the heterozygote rhodopsin heterozygote? Why they don't using rhodopsin homozygote? Why they don't using the triple knockout mice? Triple knockout mice that's the rod for photoreceptor transducing and con photoreceptor CNGN1 channel, and LPN4 are totally knockout. But the cell structure is preserved. That's auto segment, the photoreceptor. Why don't I use this system? And especially, actually, this triple knockout system is generated by the author, by this article's author. And although the author claimed that this can delay the retinal generation, but the question is that the previous state, the background, or the baseline, yes, the baseline degeneration is very serious. To me, it's not very serious. But in this study, we really need to appreciate that like the very fine design of this the whole story, because they're using like the 10 Y type control and the 10 rhodopsin heterozygote mice, the 10 to 10, a total 20 number, and it indeed save a lot of life of the animals. And this we need to like show a great appreciation for the cherished lives of the animals.